Right, so to adapt the polarity or the strength or how tightly the DNA coils to change the effect on the protein synthesis accessibility of the genes, you can add a phosphate group to the helix. So the phosphate group, or acetylation, or adding acetyl group to the histone, can decrease the positive charge of the histone. So decreasing the positive charge of the histone would mean that the electronegativity wouldn't be so strong between the polarity of the bonds, meaning it would coil less tightly because the bonds wouldn't be so strong, as they wouldn't be so attracted to each other. This would allow the genes to be easier to transcribe by the RNA polymerase, allowing certain genes to be transcribed. Methylation, methyl group is CH3. Adding a methyl group, however, to the histone would make it more hydrophobic, so it would make it bind more tightly. DNA coils tighter than preventing transcription of the gene, so it prevents some genes to be expressed and transcribed because the methyl group is more hydrophobic. So what actually is epigenetics? Epigenetics is the control of gene expression by modifying DNA. So it's the whole modifying the histones in the DNA causes to what the name epigenetics is. So if we just go up to here, we have post-transcriptional or translational control. So this is what happens on the DNA strand, on the RNA processing. So we start with a DNA strand, as you can see here. The DNA strand is filled with exons, introns, and so on. So segment, they wouldn't be so evenly spread out, but it's exons, intron, intron, etc., etc. And then the production of the production of the transcription is a precursor molecule. So after it's gone through transcription, so after we've gone through transcription, we've got transcription here. After transcription, we get a precursor molecule, which is pre-RNA. So we result now in pre-mRNA. That's our first step. So, and then later on, this pre-mRNA can be later modified to form mRNA, which is mature RNA. So it goes just in a DNA strand, transcription, pre-mRNA, which is precursor RNA, then mRNA. The processes go from transcription and then something called splicing. So, on the pre-RNA mRNA strand, it does still go exon, intron, etc., the same as before, but now we have a cap, which is a modified nucleotide. So a cap is now going to be in orange. The cap means this cap here, which is added. This cap means that it is the head of the animal, or the head of the DNA, and obviously the tail, which is at the back end, the tail is added to the other end. These ends, so the cap added to one end is added to the 5 end, and the tail is added to the 3 end. So that just goes in order. So cap, so say you've got a little person, the cap on there, someone's head, someone wearing a cap, is going to be the highest because they're at the top, be the biggest number, the 5, and then the tail at the bottom end is going to be the 3 because it's at the bottom of your bottom. So it's 3. Also 3 looks like a bottom, so at the end, at the 3. This this cap and this tail allows stability to the mRNA and delays the degradation in cytoplasm. So I will go over that in a second. The addition of the cap and the tail allows for two things. One, it allows for stability. And two, the cap and the tail allows for delay in degradation in the cytoplasm. Delay, so basically a longer lifespan in degradation in cytoplasm. So the cap and the tail allows it to stay in the cytoplasm for a lot longer. The splicing, which is the second mechanism, the splicing occurs where the RNA is cut at specific points. So now we get to the mRNA, which is the mature RNA. Here we only have exons. The introns that were before have gone. So we go intron, exon, etc. on the DNA. Then we go cap, exon, intron, tail. And then after splicing, the third and final strand, we just get only exons, but we still have the cap and the tail, obviously, to keep the delay in degradation and the stability of the DNA. So the cap and the tail are still there. But we only have exons this time. The mature RNA, which is the last one here, this combined to the ribosomes and therefore code for the protein, which these two can't. It can't code for a protein, and we'll get to that in 1 second. So this splicing, which is the last step, actually occurs where the RNA is cut at specific points. So the RNA is these molecules here. The RNA is cut at specific points, so the introns, which I talked about earlier, these ones, these introns are removed because we don't want the introns. And the exons are therefore joined together, the removal of intron DNA. Both the splicing and the cap and tail addition occurs within the nucleus. So I always do the nucleus in green for some reason. So it always still occurs in the nucleus before leaving the pores. This is always in the nucleus. So why don't we want the introns? The introns do not code for DNA, as exons codes for DNA, and exons which then can code for polypeptides, which are then copied and sent to ribosomes. So we just have to remove those introns through splicing, because they're no use to us. They don't do anything. It's just wasted DNA material. The splicing, which is an important step here, this is catalyzed by something called the spliceosome. So splicing is catalyzed by the spliceosome. The restriction enzyme is the enzyme that actually cuts the introns off of the RNA and into and out of the cytoplasm. So the restriction enzyme, single strand of base is then left. So restriction enzyme, and we do some scissors because the restriction enzyme acts as the cuts, and it cuts the introns out. But the restriction enzyme actually leaves something called a sticky end, and the sticky end allows the exons to then form and bind together and stick as such together with the positive hydrogens that are left. So RNA editing, this is just recap of the ribosomes, what a ribosome looks like. They're on the rough RER, so on the rough endoplasmic reticulum. They are the cell's protein factories, basically. They make and package proteins, and they're on the endoplasmic reticulum, around the nucleus. Just a reminder there. The nucleotide sequence can be edited, changed and edited by base addition. So I've actually got a new one here. So, this is my new poster. The nucleotide sequence, which we just talked about, no different, can be changed by base addition, deletion and substitution. So, to change the sequence of the base or RNA is addition, basically just like year 7 maths. Addition, deletion and substitution. That's fairly obvious. The only one we're missing is multiply. So addition, adding a gene, deletion, taking a gene out, and substitution, swapping genes over. So adding, deleting and substitution. This is the same effect of a point mutation